Welcome viewers. Today we are going to discuss on pure culture techniques. In the field of microbiology, we are very well acquainted that we are residing in a universe totally surrounded by microbes. Microbes are present everywhere in the universe, whether it be soil, water or air. But the question arises that the study of microbes can be made possible only when we cultivate them under laboratory conditions. To cultivate them in laboratory conditions, we need certain culture media. In our earlier discussions, we have dealt in details the various varieties of culture media that are available for the growth of microbes. Merely growing them in culture media is not enough to study the microbes, because if we just try to deal with a mixed population of bacteria or mixed population of microbes as they reside in the universe, if they are residing in a media together, then we can have only idea about their morphology and their outer structures and all these things. But if we want to study precisely their growth parameters, their various different mechanisms of establishing themselves in their suitable environments, then we really need to have a pure culture.
Ma'am, what is the utility of pure culture in microbiological study?
Yeah, in microbiological study, we need to have a pure culture, because by getting a pure culture only, we can have a detailed idea about the nature of the microbe, its method of multiplication, its method of replicating its genome. And if we have a pure culture, then only we can maintain it properly, and we can have various techniques to enumerate the number of microbes which are present in a particular population. This can only be done if we do have a pure culture in our custody. The development of pure culture was not a very easy journey. It was a very difficult job, and it needed an enormous amount of effort from various eminent scientists all over the world. So let's have a look onto those scientists who have contributed sufficiently in developing pure culture techniques. So the first name that comes in our mind is the German bacteriologist Robert Koch. In the year 1881, he started working with boiled potatoes. What he did, he made a slice of boiled potato with sterilized knife, and onto those sterilized potatoes, he used that surface of sterilized potato for streaking purpose. And by streaking the mixture of bacteria, he was able to isolate single colonies. So this was the first effort to develop pure culture by using boiled potatoes.
Ma'am, what was the drawback of using sliced potatoes for obtaining pure culture?
Yeah, the major drawback with boiled potatoes was that that potato was used as a source of nutrient for the growth of bacteria or for the growth of microbe. So all microbes won't be able to utilize the nutrients present in the potato for their growth. So all types of organisms won't be growing on the potato. So we won't be able to get hold of actually all varieties of microbes that were present in the mixture. So that was the major drawback, right? So then what Koch did, that he used gelatin as a solidifying agent. He started his work with Loeffler media. Loeffler media was actually meant for the growth of pathogenic microbes. Actually, Koch was very much interested in cultivating pathogenic microbes, because by cultivating pathogenic microbes, they can be studied properly, and then only we can establish methods of their growth, then only we can restrict their growth. Until and unless we cannot study those pathogenic microbes, we cannot design drugs for their control. That's why Koch started his work with Loeffler's media, which comprises of meat extract and peptone. Into it, he added gelatin as the solidifying agent.
Ma'am, in our laboratories, ma'am, why don't we use gelatin as the agent? Why do we use agar instead?
Yeah, the major drawback of using gelatin is that gelatin gets liquefied at 37 degrees centigrade. And 37 degrees centigrade, or the physiological temperature, is supposed to be the most suitable temperature for the growth of pathogenic microbes. So utilizing gelatin, actually we are unable to grow the pathogenic microbes. That's why scientific community was hunting for a better solidifying agent over gelatin, and we finally ended up in agar. And actually the discovery of agar as a solidifying agent has emerged from a very interesting discovery. Therein what we have seen, that a Japanese innkeeper, Minoru Terazaemon, what he could find, that once he threw away some seaweed soups, right, and on the next day he find that the seaweed soup has solidified because of the cold temperature. Then it was found that agar, which is isolated from seaweeds, can be used as a solidifying agent. This suggestion was basically given by Mrs. Hess, wife of Walter Hess. Walter Hess was actually a helper in Koch's lab, so he was often discussing that he is looking for a good solidifying agent, and his wife then suggested him that why don't you use agar as the solidifying agent. So this was a remarkable contribution in the field of microbiology, that we got hold of agar and we now every time use agar as the solidifying agent. So the major benefit of agar over gelatin is that it remains liquid in the temperature of 90 to 95 degrees centigrades, and it solidifies under 40 degrees centigrade. So it remains solid even at 37 degrees centigrade, which was the major drawback with gelatin, right, because it used to liquefy at 37 degrees centigrade. And the second benefit of using agar was that it cannot be utilized by the microbes. As gelatin was utilized by various microbes as a source of nutrient, so it was broken down in presence of microbes, but agar was not a source of food for most of the microbes. Now let's go back to various methodologies which came gradually that helped us to develop the pure culture techniques. So first of all, we will talk about Emil Hansen. He was the first person who used methodologies to get pure cultures of yeast. His methodology of opting pure culture was just visualizing the microbe under the microscope, and he kept visualizing it until he got hold of a single microbe or a single yeast cell that finally developed into a colony. But this methodology was not very suitable for getting pure cultures of bacteria. The next name that comes in our mind is Barber et al. What Barber et al. Did, that they picked up each and every single bacteria that they found in the hanging drop experiment. Right? Say for example, if there were 4 different bacteria present in the hanging drop broth, then they inoculated each and every bacteria in a separate broth. This method was very nice to work and to get hold of a pure culture, but there were certain limitations, as the isolation of bacteria from a hanging drop was a method that required various intricate apparatus, number one. And number 2 drawback was it was very difficult to get hold of a single bacteria utilizing such instruments like Pasteur pipette or inoculation needle. So this was the drawback with Barber et al.'s method. Next method that was of Buri's India ink method. What Buri did, that he made an emulsion of bacteria in diluted India ink, and he used this diluted suspension for spotting on gelatin containing petri dishes, right? And what he did, from each spot that he got on the gelatin petri dishes, he visualized those spots through microscope on dry slides, right? Then from those slides wherefrom he got single colonies or single bacteria, he spotted those bacteria and utilized or allowed them to grow in another culture, or he subcultured them. And subculturing methodology was to take out that spot wherein single bacteria was there with the help of say some other slides or some cover slips. He used certain cover slips and he placed the cover slip on the top of the India ink spotting that he did on the petri dish, and that cover slip was then utilized for inoculating into a broth culture. So this method was very nice to get hold of pure culture of bacteria, but there was again a drawback that there are certain bacteria which couldn't survive in presence of India ink, right? And the second drawback was it was very much difficult to place the petri dish on the stage of the microscope and to visualize the different spots. So removing the stage of the microscope was a tedious job by placing the petri dishes on the top of the stage. So this was then overruled by Orskov. What J. Orskov did, that instead of petri dishes, he utilized glass slides, and onto the glass slides, with the help of pass-through pipette, he added the gelatin. Right? After addition of gelatin, the rest of the method was same, that the India ink containing bacteria was spotted and the stage was moved gently to get hold of the spot which contains a single bacteria. And this method was a better option to get hold of pure cultures. There are contributions by many other scientists. The list is endless, but we have got limited time, that's why we restrict ourselves here. Nowadays, we generally use 3 different methods for getting pure cultures. Number 1 method is spread plate method. Second method is streak plate method. And the third method is pour plate method. Now let's start with spread plate method. Utilizing spread plate method, we can have an estimation of number of microbes present in a suspension. In this method, we start our experiment by taking a petri dish. Into that petri dish, first a nutrient agar is poured. It means it comprises of a nutrient which is solidified with agar. Onto that plate, we are going to pour diluted suspensions of microbes. For that, it should be taken into account that per plate there should be in the range of 30 to 300 bacterial colonies. So as we don't know about the actual number of microbes present in the suspension, we need to go for a serial dilution method. What do we understand by a serial dilution method? We take sterile test tubes. Into those test tubes, we add equal amounts of sterile water. And the suspension where from we are trying to calculate the number of microbes is first added on the tube number 1. And gradually we take suspensions from tube number 1 to 2, and by doing so we are diluting the microbial suspension 10 folds, and further we move it to the next tube, then we are diluting it 100 folds. So in this way we can make a serial dilution of 10 fold, 100 fold, 1000 folds and so on. So once we do this type of dilutions, we can carry on the method of spreading. We will take say some amount of 10 or 100 or 200 microliters of suspension, and we will pour it on the middle of the petri dish. Once we add the bacterial suspension on the middle of the petri dish, then we will take a spreader. What is a spreader? Spreader is a glass rod which is bent in 90 degrees, and with the help of a spreader we will spread the entire microbial suspension that was present in the center of the petri dish. By spreading, we make it sure that the bacteria are spread uniformly onto the agar, so that each and every bacteria gets ample amount of space for its growth. It is considered that one colony corresponds to one bacteria, that each colony that we can see in a spread plate method has started its journey from a single bacteria. So just by calculating the number of bacterial colony that we are getting in a spread plate method, we can estimate the number of microbes that were present in the bacterial cell suspension. Obviously, we should take into account the dilution factors. If we get too many number of colonies, say for example in the range of 1000 or 10,000 colonies in one petri dish of 90 mm, then what will happen? That the bacteria won't get enough space to grow properly, as a result of which the size of the bacterial colony will reduce and they might merge together. So this will be difficult for us to calculate the number of bacteria. That's why, as I said earlier, that it is essential that in a 90 mm petri dish, at least 30 to 300 colonies are present. Even lesser than 30 is not good for calculation, or more than 300 is also difficult to calculate. So this is all about spread plate method of getting pure cultures. Next we'll start with streak plate method of getting pure cultures. Streak plate method can be of three different types. One is called direct streaking, second is called sector streaking, and third is called four flame streaking. So let's start with the different steps of streaking methodologies. In this method, we start our journey with a culture suspension. Usually, this method is to be opted when the microbial titer is less. Under such circumstances, with the help of an inoculation needle, which there is a nichrome wire attached onto the inoculation needle. What we do, that we first heat the nichrome wire in the oxidized flame and make it red hot. This is done to make the nichrome wire sterile or free of microbes. After that, the nichrome wire is cooled down and that is introduced into the test tube that contains the microbial suspension. Once we insert the nichrome wire inside the test tube where the bacterial suspension is there, a loop of culture is taken out and it is streaked onto the agar plates randomly, right? With continuously we spread it, and as we spread it continuously, as we are moving away from the point where we started streaking, we are gradually diluting the bacterial suspension. So the spreading is done basically to reduce the bacterial titer, and as many strokes as you put, you are gradually diluting it more. So once you keep on streaking it throughout the plate, say 50 to 500 strokes are made, and finally they are kept inside the incubator. The next day we will see that one end of the petri dish where we actually terminated the streaking, we get discrete colonies. So this methodology helps us to separate out bacterial population that were residing together in a broth culture. The second method of streaking is called sector streaking. During sector streaking, what is done, that the petri dish is taken, it is inverted, and the circular petri dish is divided into 4 equal sectors, right? So two lines are drawn diametrically and we get 4 different sectors. So if I start streaking with one of the sectors, once I end up into one sector, I will put a tail, I will extend the streaking, and once I terminate there, after that I will start the streaking of the next sector from the tail, right? So the purpose is we are gradually diluting the bacterial suspension or the microbiological suspension as we move from sector 1 to 2 and similarly from 2 to 3 and 4. So the most least populated bacteria will be there in the fourth sector. In the fourth sector, we can expect to get pure bacterial cultures, right? This is supposed to be the second method of streaking. The third method of streaking is a bit similar to the second method, 4-flame streaking. Just the difference is, once I complete streaking of first sector, I will again flame the inoculation needle in the oxidized flame, and thereby I will reduce whatever bacteria that were present onto the inoculation needle. Then I will again cool it down, start taking the culture from the tail of the first sector and again streak on the second sector. So this process continues. The second method of, say, sector streaking or 4-flame streaking are a bit similar, but just the difference is that I am flaming after completion of streaking in each sector. So basically, streak method is used to get pure cultures if the culture is not heavily loaded with microbes.
Ma'am,
Yeah.
among streak culture and spread plate culture, like which one is used to get pure colonies?
Say for example, if you are working with environmental samples where you exactly don't know that how many number of microbes or how many varieties of microbes are there, then it is always suggested that you go for a spread plate method by going with proper dilutions, right? But this streaking method is generally used, say for example, if in your lab 2 or 3 cultures got contaminated, or it is a mixed culture of 2 or more bacteria, and you want to separate out at least 2 to 3 different bacteria, then you go for streaking, because streaking won't help you to reach to a pure culture if there are many microbes together residing, right? This is what I consider. Now, the third method of getting pure cultures is pore plate method. Pore plate method also, what we do, we carry on the serial dilution of the bacterial suspension whose actual titer is to be determined. Serial dilution method we discussed earlier. So we have got a serially diluted tubes containing different bacterial suspension at different dilutions, right? Then what we will take, we will have a test tube that contains a nutrient agar at a temperature of 40 to 45 degrees centigrade. Into that agar media, we will add say 200 microliter of bacterial suspension from each of these 3 or 4 different diluted samples of bacteria. Then we will immediately pour it in a sterile petri dish, and within the petri dish, the media solidifies. The bacteria that was inoculated along with the nutrient agar also starts growing, and it will give discrete colonies. So in this methodology of pore plate technique, what we get, we can get colonies of 3 different varieties. Some bacteria will grow on the upper surface of the agar media, some of them will grow in between, and some of them are growing beneath, means some of them are just under the surface of the agar. So 3 different varieties of colonies can be obtained in this pore plate methodology. This pore plate methodology is a very good option for cultivating the bacteria, but there is one problem lying with pore plate methodology, that when we consider the number of bacteria that we get as colony forming units, right? We don't consider them as colonies that are emerging from single bacteria. The reason behind that, whenever we pour the agar media containing the bacteria onto the petri dishes, there are chances that some bacteria might remain attached together. Say for example, 2 bacteria remaining attached together, and both of them are jointly giving 1 colony. So it is better option that whatever colonies that we are getting by pore plate method should again be checked by going with a streak plate method. So if it contains still more than 1 type of bacteria, then we can purify them by streak plate method. So in our lecture on pure culture techniques, today we have discussed basically the contributions of eminent scientists that helped us to get hold of pure culture nowadays, and actually that development of pure culture has changed the microbial world, or study of microbiology has become very much easier, and now we are in a position to cultivate various pathogenic microbes, and we know their mode of replication, their mode of existence, and how we can control their growth has been possible to study because we are able to culture those microbes, right, under pure conditions. So in all, we have discussed 3 different methodologies to get pure cultures. Number 1 method was streak plate method, then we have gone through spread plate method, and finally we discussed about pour plate method. Depending on situations or the number of microbes present in a sample, we will change the mode of using pure culture methods.